at graphically how there's a tiny, tiny amount of our DNA that actually codes for proteins or for RNA. Right. There are also these things in our DNA that are called transposable elements, and these are very interesting because they're not genes, but they're pieces of DNA. They don't code for anything, but they're pieces of DNA that move. They jump, literally, from where they are located to a different location. And they might jump to the same chromosome. They could actually jump to a different chromosome. And when they do that, they often have some pretty devastating effects on whatever gene because they can jump into the middle of a gene sequence. And if they jump in the middle, then that gene is no longer going to work. About 75% of all human DNA is made up of transposable elements and sequences related to them. So that's a lot of, and we still don't really know. Barbara McClintock back in the, was it 80s, I think? She worked at University of Chicago, and she realized that these things existed, but we still are trying to figure out what these mobile pieces of DNA do, and we're not really sure. All right, you want to really pay attention for these next slides. Ready? Ready for it? X. Taking it out. Taking it out. No, this is not a fool's joke. Not unless you want it to be. Everything up until 21.5, you can cross those slides out. Okay? We're not going to talk about some transposable DNA any more than that. We know some things, but not that much. All right. So the basis of the genomic level for the change in the genomic level. So your genes, why do they change? That's mutation. And we can make the assumption that early on, whatever genes you had, you used everything, right? There wasn't any extra. And then at some point over time, things became eliminated. There were mutations. Now they didn't work as well. And so now we have these very large genomes that have a lot of genes in them. And they don't necessarily do anything, but they provide us, like I said earlier, they provide us with this raw material. So now we have this sequence, and it is possible for a gene that didn't work to now get back into the flow and become something that works because now a mutation does something else, and all of a sudden it can be turned into, that recipe can be used for something else, whereas it wasn't being used at all for a long time. When we talk about this change in genome, we can talk about things like accidents and meiosis. And accidents and meiosis can lead to all sorts of things. One of them that it can do, we've already talked about, which is polyploidy. So that's where, you go from having, 4 sets of chromosomes to now you have 6 sets of chromosomes. An entire extra set of chromosomes is copied rather than just having a tiny piece of a chromosome copied or one gene. And that can have huge changes in the DNA or in the function of the genes in a species. Now, the other thing is, if that happens, if you go from 4 sets of chromosomes to 6, you can no longer reproduce with that set of 4 chromosomes because now you're going to have a mixed up number and the body's not going to know what to do with that number. So normally that polyploidy only happens in plants rather than in humans. Plants seem to be a little bit better at tolerating them for one reason, because plants can reproduce asexually. So they don't need a partner. If they don't need a partner, then having an extra set of chromosomes wouldn't be that big of a deal. I always like to talk about this one because it plays to the idea of how change in chromosome number could lead to a different species. And I'm just going to, I think I'll just show you the picture here. So in this case, if we look at, I always forget how many chromosomes there are in each. Humans have 23 pairs of chromosomes and chimpanzees have 24. And so a lot of times people will say, well, obviously we're not very closely related, or we're not related to chimpanzees because they have a different number of chromosomes. But what's interesting is if you actually look at the chromosomes, you see that what appears to happen is two of their chromosomes, specifically, and you don't need to remember the number, but specifically chromosome 12 and chromosome 13 appear to have joined together. So these guys have joined together because if you look at our chromosome number 2, we have an area in the middle of the chromosome, which makes no sense. That looks like, remember how we talked about telomeres? Remember what telomeres are? What are telomeres? Yeah, they're the extra stuff. Remember how we talked about how how DNA polymerase has to have something that it can attach to. Well, when you get to the end of the chromosome, you can't do anything with that. So the body has evolved a way of dealing with that by putting on this area, this telomere area that's just repeating sequence that seems to not ever code for anything, and it doesn't matter if you cut it off. It does seem to tell you how old the chromosome is and how old the cell is, but it doesn't seem to play a role in anything else. Well, if you're cutting off the end of the chromosome, that's one thing, but you wouldn't want to ever cut off an area right in the middle because that would be pretty detrimental. So that doesn't make a lot of sense. So we see this telomere-like region, and if you look at the genes that are located on R2 and you line them up with the ones that are on 13, they're very similar. I mean, obviously the sequences are different, but they are coding for the same proteins. So they fuse together. So in this case, we have an example of how now we went from one species to now we had two different species. You can also look at sometimes little pieces will break off. If you look at the human chromosome versus mice, there are big chunks of DNA that correspond on R16 to the mice. So this chunk seems to have broken off and traveled to a different chromosome. So that's another example of that happening. And these can happen. These chunks can happen during meiosis. You can have a duplication that gets transferred. You can have something move to a different location. You can have something be inverted, so it's upside down. And by looking at these different segments, we can kind of then get some ideas about not only the history of our species and the chimpanzees or mice or whatever you want to do, but you can also start to look at why these areas might be important. It's like, whoa, I didn't even know that this gene was important, yet mice have it, we have it, rats have it, chimpanzees have it. It seems to be conserved. So we talk about this idea called conserved. And that just refers to the idea that it's there in every species. It's conserved through the different species, and that seems to play a role in its importance. Duplications and inversions seem to have accelerated about 100 million years ago, which also corresponds to the time when dinosaurs went extinct. And at the same time, mammals started to become very diverse, and there was an explosion of different species of mammals that we hadn't seen before that. And so there were probably these duplications and inversions before, but they weren't successful because at that point, the environment wasn't successful. So it wasn't until the environment was ripe for a change, in this case, the Earth getting cold, the reptiles not being able to survive, and the mammals being like, yeah, I can do this, that those duplications and inversions tended to be more successful. Sometimes you get, we talk about it all the time. I talk about how crossing over, when it occurs, there's a one-for-one change. So what happens during crossing over? Let's just talk about that for a second. What happens during crossing over? Trying to bring back all those things that we talked about. Yeah, so you have homologous chromosomes, and those homologous chromosomes will overlap, and they will exchange genetic information, which is great because it diversifies each of the chromosomes. It makes the chromosomes different from what the parents look like. However, and that usually happens a one-for-one. So if I have 20 genes cross over, 20 will be given back. But sometimes it isn't equal. And so you can have a deletion where certain genes or even a chunk of a chromosome disappears. Also, these transposable units that I talked about, sometimes those will jump during meiosis. So the chromosomes are all lined up, and they will jump, and they don't necessarily jump to another chromatid. They will jump to a homologous chromosome. And so now, all of a sudden, in that particular homologous chromosome, the genes are going to be incorrect, and they're not going to work the same way they had before. Okay, when all this happens, most of the time, we think, it results in things where nothing works very well. But occasionally, you get genes that have novel functions. So as one of the things we talked about was this gene duplication. So sometimes genes get duplicated, and sometimes those genes get mutated out of function, so they don't work anymore. Occasionally, they get duplicated, and they're not in the same location anymore, right? They're in different locations on the gene, and that means they're going to be exposed to different environmental conditions. So a UV ray could hit it and cause a change in that gene sequence in a way that's novel from its partner gene down the way. And there's an example of that with the lysosome gene. And the lysosome gene was duplicated many times, but the lysosome gene can sorry, I'm saying lysosome. Lysozyme gene can do a couple of different things. Its old form is it will protect against, it produces a protein that's actually an enzyme, and that enzyme helps to prevent bacterial infections. And obviously, infections are, any kind of animal can get an infection. So it's kind of a generic work against bacteria kind of role. But then another gene has changed its structure and its DNA sequence. And because of that, it has changed to what's called alpha lactalbumin. And what that does is it helps to produce milk, okay, in mammals. So it went from being kind of a multi-purpose gene that worked for fighting off bacterial infections to now it is helping with production of milk in just mammals. Okay, so its origins are as what's called lysozyme, but it's changed to be something else. There's a picture of this in the next page showing the two different outcomes from the same genetic sequence, or from a similar genetic sequence, I should say. So it's lysozyme and alpha lactalbumin. And anything that's in yellow here represents represents the same amino acids that are being made. So all these amino acids have, the 20 different amino acids have different letters that represent the amino acid. That's why you see a single letter like an S or a G or Y. They all represent different amino acids. And anything that's not yellow here is going to be something that's different. So they're significantly different, but they're significantly alike as well. And they have a common origin. All right. Let's see. We're almost done. So comparing genomes can be used for all sorts of ways to look at things like evolutionary history, how closely related things are to each other. But they can also be used for things like, okay, if this gene is found in all of these different species, that indicates that it's probably a pretty important gene. Because if it wasn't, it would have been mutated, it would have been out of function by now, and it wouldn't have stayed around in all of these different species. So highly conserved genes have changed little over time, and that helps to clarify relationships between species that diverged a long time ago. We can look at bacteria, archaea, and eukaryotic domains, and we see that they diverged from each other between 2 and 4 billion years ago. But highly conserved genes can be studied, again, because, this is another good point. Not only can you know that gene is important, but you can also then, if you study it in one organism, and it's mostly the same in a different organism, you can apply whatever you learned about that gene in that particular species, you can apply it to the other species, and you know that it probably has similar implications in that species. I actually had a student a long time ago that she loved theater and she loved, I guess, biology. And she was a double major at WashU in like theater craft and biology. And she ended up getting a PhD somewhere on the East Coast it was just so like, it was way over my head, but it's basically genomics. And so she studied a couple of different species genomes, and her PhD was on the similarities and differences between them. And I know I'm not doing justice to what she did, but it's definitely a species that, it's definitely a field that's growing, and a lot of people are getting involved in. Genomes of closely related species are likely to be organized similarly. And that is important because now we can figure out, okay, so there's all this genetic information, but for a lot of it, we don't know really what it does. We don't have a very good idea of what it does. So, if we look at two different mammals and we see that they have this conserved area that's very similar, and therefore probably does the same thing, it gives us clues to, for example, what makes a mammal versus what makes a cold-blooded ectotherm. Just wanted to point that out. And that's not really all that important. Let's see. All right, let's see. I think I'm just going to leave that at that. Okay, last two are out. Okay.